We're going to go over messed up mitosis, the cell cycle, cancer, and nanotechnology, and this is the background information that you need as teachers. This is the cell cycle, just a broad overview. We're going to break down each of these stages so you know more about it. In interphase, it's all of this first bit. It's the majority of the cell cycle. So in G1, it's called gap 1, there's cell growth, and the ribosomes and proteins are duplicated. We move on to the S phase, then you have the DNA and the centrosomes are duplicated. Then next you have G2, gap 2, and the organelles replicate. So after all of this, now we're into prophase. Prophase is the first step of the mitotic phase. So I don't want to beat you to death with the information, but basically what you need to take away is the cell is getting ready to divide. So this nuclear envelope is going to go away, and then we get centrosomes with the microtubules. In metaphase, the chromosomes line up in the middle of the cell along the midline, and they're going to be ready for anaphase. In anaphase, the sister chromatids get pulled apart into the separate ends of the cell. And then in telophase, the nuclear envelopes start to reform around each group of the chromosomes, and the chromosomes become less condensed. So you're really starting to get two different cells here. And then the last bit is cytokinesis, and that's when this cleavage furrow that's right here in between the cells is going to form, and you have the two cells are going to break apart and pinch off, and so you have cell replication. So as we're going through the lesson, we start off with mitosis in the cell cycle, and when that gets disturbed, that leads to cancer. Cancer is unregulated cell division, and these cells just keep dividing, and there's no input from the cell. The DNA has been corrupted, so there's no input from the cell to stop this division. And so this is how you get tumors, and this is why cancer is so nasty, unregulated cell division. And so we want to hit 3 common types of cancer treatment that are available. And with chemo, what's going to happen is they inject drugs into your bloodstream, so it's going to target the whole body. They use this for when cancer has metastasized, and that just means cancer originates in one spot, and the cells can break loose and go through the bloodstream and attach somewhere else and start a new tumor. So when cancer starts to spread throughout the whole body, that's when it's metastasized. So chemotherapy is really good for this, because sometimes you can't tell exactly where the cancer has spread to. You can maybe see the original tumor, but you can't see the rest of it. So chemotherapy will target the whole body. It uses drugs. And one of the unfortunate side effects is that it damages healthy cells. It damages the hair cells, and that's people that are on chemo. That's why they go bald. Radiation they use at just one spot. They can target one specific spot. They use electromagnetic waves. However, this damages some of the healthy cells that are nearby. And then surgery, obviously, they can cut out a tumor. They'll use a scalpel and just cut it out, but they have to make an incision on the surface and go prying around in your body, and it causes bleeding. Again, healthy cells get damaged. So there's this kind of this issue here where they're all damaging healthy cells, and we're trying to figure out something that maybe won't do that. First, we got to talk about nanotechnology real quick. Nano is just a prefix for 10 to the negative 9. That's 1 billionth. So if we're talking about meters, a nanometer is 1 billionth of a meter. It's incredibly small. It's so small you can't see it. Here are a few just fun little facts about nanometers. My favorite is that your fingernails grow 1 nanometer per second. It's just, you can't see that. These carbon atoms, 5 carbon atoms end to end is 1 nanometer. So we're talking something that's really, really tiny. And as you're going through, on your version of PowerPoint, you can click this nanoscale, and you can just drag the slider in and out, and it will zoom in and out on objects, and you can see how small nano really is. So one of the fascinating things about nano is it's so small that it's starting to be affected by quantum physics as well as classical physics. So you get these varying here when we're looking at, well, obviously A, B, C, D, and A, B, C, D are all gold, but in E, F, and G, that's also gold. What's happened is we have different sized gold nanoparticles in a solution, and depending on the size, you get these different colors. They actually did this with stained glass way back in the medieval times. This is how they got the different colors on the stained glass. They used nanoparticle gold and silver. Of course, they didn't really know that, but they just knew that they got different colors with these different solutions. So the properties, this is the big point, is that the properties of materials are different at the nano level than they are at our level. So with color, modern sunscreen has nano-sized zinc oxide in it, and that's what makes it transparent. You can, in quotations, rub it into your skin. So the old sunscreen that you see on people's noses and whatnot it was macro-sized zinc oxide. So still small, but big enough that it's not transparent. Another example of color is that these new quantum dot or ultra HD televisions that are on the market, they use nanoparticles in the screen itself, and so when they're excited by the electrical current, they actually light up the whole color spectrum, and not just like the old pixels or just the primary colors. And there's more. They're putting nanoparticles for strength. They're putting them in your car bumpers and baseball bats and hockey sticks. The magnetic properties are different, and then conductivity. Some nanoparticles are highly conductive. This one photo here is that's a nanowire wrapped around a human hair being excited with a laser. So super conductive, and that's the one that we're going to home in for this lesson, is the conductivity. So nano-sized carbon structures, you know graphite, you know diamond, and that, you know it's just carbon arranged in a different lattice here. So there are fullerenes, and we're going to home in on the nanotube. We have graphene off to the side because if you look at the structure closely here, graphene is the same thing as a nanotube, except it's just not rolled up. So again, if you look at graphene and then come back over here and look at graphite, it's the same hexagonal structure. Graphite just has layers. So graphene is just one sheet of graphite. And then a nanotube is graphene rolled into a tube. There are two types of nanotubes, single-walled and multi-walled, and it's really just simple as that. In the multi-walled, you got multiple nanotubes that are inside of each other, and single-walled is just one. So now that we've introduced nano and how its physical properties are enhanced in a way, we're going to go ahead and try to apply this to our cancer treatment. So part of the thing about cancer cells is they metastasize, they spread, they vary in size and shape, and they're hard to get rid of because they're in with all the healthy cells. And all these treatment options that we have damage the healthy cells as well. So we want to destroy cancer cells that may have spread, and we want to treat those cancer cells without harming your healthy cells. So we got to get to them, we have to target specifically them, and then we have to destroy them. That's the challenge. So using nano to search, what we're going to do is, since it's so small, we can put it in a vial and inject it into your bloodstream. So if you look here, the size of this red blood cell is micrometers. So a nanotube is far smaller than that. So they can get in the bloodstream, no problem. And then cancer cells, one property that they have is they have this leaky vasculature. So stuff gets in them anyway. And then one of the keys to this lesson is they have overexpressed amount of folate receptors. Now this is in some cancer cells, the different types of cancer cells. So this treatment isn't perfect for all cancer cells, but we've done research and there are different kinds of receptors and different kinds of cancers that are overexpressed. So really, this can work to your advantage, but to keep it consistent, we're just going to talk folate. So the overexpressed folate receptors, and all these receptors do is accept folic acid into the cell. So what we can do is we can put folic acid, we can coat our nanotubes in folic acid, and then they will bind to the cancer cells. So we talked about the conductive property of these nanotubes, and this is getting into physical science, but we need to swing by and make this stop. We're going to use near-infrared radiation, which is part of the visible spectrum. It's near-infrared, not infrared. So we're talking on the red end of the visible spectrum here. That is transparent to the human body. It doesn't harm you at all to have red light shine on you. So, but it can penetrate into the tissue. So what we can do is we inject these carbon nanotubes in the bloodstream. They bind to the cancer cells because of the overexpressed folate receptors. And then we can shoot a laser at the entire body, since it doesn't harm your healthy cells. And it causes these nanotubes that are highly conductive to heat up. So it's just the fact of energy transfer, okay? The wavelength of the light excites the carbon nanotube and it begins to heat up, and that heat will then conduct into the cancer cell. So the cancer cell is heated up, and then it dies. You heat something up enough and it will die. So this chart, as I was saying earlier, the percentage of folate receptor overexpression. So in ovarian and endometrial cancer, this is a huge option. Like I was saying, we're talking about folate receptors, and in breast cancer, there's other types of receptors that are overexpressed. So you could coat your nanotubes with something else. But again, we just want to stay consistent and talk about folate receptors. So part of the disadvantages or challenges in this treatment is we got to think about what happens to these particles after they destroy the cells. Your body can essentially break down and recycle the dead cells, but is that the case with the nanotubes? And since this is so new in the research world, we're not exactly sure. And what effects can these nanoparticles have on your body? Do they float around your bloodstream and get in your brain? Or there's been some research done, and scientists disagree, but some scientists say they can aggravate the lungs like asbestos does. I mean, these are just tiny fibers. Of course, there's disagreement on that research, but it's something worth looking into. You know, what happens if my body doesn't break them down? They get in my lungs, and now I'm breathing them all over my family members. Or what happens when I pee them out and they get in the water system? So it's just good things to think about. These are other treatments. Since the asbestos stuff came out, nanotubes kind of got put on hold. But so they're looking at gold nanoparticles. They call them nanoshells. So same thing, you inject it into the bloodstream, they get to the tumor, they do this receptor coating, the nanoshells go into the cell, you hit them with near-infrared radiation, they're super conductive, the water in the cell evaporates and it makes it pop. And so this is really cool. They can actually hear this pop and then know where the cancer is that is metastasized that you weren't able to locate beforehand.